The next few talks are going to focus around antimicrobial resistance, or AMR as we call it. This is an emerging problem for veterinary medicine and human medicine. And the solutions are quite complicated because of the interaction between so many different sectors that are driving this problem. So today, my name is Sophie St-Hilaire. I work in Hong Kong. I'm a veterinary epidemiologist, and I focus on aquatic animals. So I'm the first speaker of the session. I'm also the moderator, so hopefully I don't go over. I'm going to share with you what we've done in Hong Kong with our aquaculture industry. If you want to put my thing up. So yeah, what we've done in Hong Kong to try to address this problem in the aquaculture industry. All right, so antimicrobial resistance is from antibiotic use, or one of the primary driver of it is antibiotic use. And so what we tried to do in Hong Kong is focus on the management strategies that are used by aquaculture that would be considered misuse of antibiotics. And we thought if we can at least deal with those, then we will progress to other control strategies. And I put up here some of the issues that we consider misuse, and I've grouped them into three things: overuse of antibiotics, inappropriate use of antibiotics, and improper use of antibiotics. And I'm just going to highlight what these differences are with aquaculture. So inappropriate use of antibiotics is when somebody uses an antibiotic to treat something other than bacterial problems. And they don't work for non-bacterial issues. But unfortunately, because fish mortalities kind of all look the same, especially to the untrained eye, some people will use antibiotics to treat water quality problems, viral problems, parasite problems. And these products are, so when you have a fish mortality on a farm, especially when you have a small-scale farmer, they don't have a lot of experience with this sort of thing. They don't have specialists on site. And there's a little bit of a panic. So they might have antibiotics that someone's given them, their neighbor or somebody else, and they'll throw things at the problem. Antibiotics are relatively inexpensive. They don't harm the fish usually at the time that you're giving them, and they're relatively easy to obtain. So if you have nothing else and half of your farm is dying, you're probably going to try it. So if we could only treat bacterial diseases, I believe we could reduce the use of antibiotics on farms quite a bit. Unfortunately, and it requires some diagnostic capabilities when you have a fish mortality event. And our small farmers have limited access to labs or fish health specialists, and they have limited funds. So there lies one of our issues. Overuse of antibiotics. For this, I'm referring to the fact that the antibiotics are used for bacterial problems, but they're just used quite a bit too often, and maybe they don't always have to be used if we had prevention strategies. So a common scenario for us, and I think for other small-scale aquaculture industries, is we'll have a water quality problem, and that's not really detected, and so that stresses the fish. That then leads to endemic parasites kind of increasing in numbers, also maybe not picked up by the farmer because mortality is still fairly low. And then eventually you get these secondary bacterial infections from common bacterial organisms, the Vibrio, the Aeromonas, those types, Streptococcus even, in the water body. So if we caught the disease earlier in the process, we would actually reduce antibiotic use. And that actually requires that the farmer has some kind of surveillance system in place where they can detect the water quality problems when they happen, or they can start to see increases or changes in the parasite loads. Again, small farmers, they have limited staff. There might be one guy on a farm, and Hong Kong is very small farms, very small industry. And they don't have the technology. They don't all have the fancy DO meters and the spin touch machines so it is harder for them to pick up these things. The other overuse of antibiotics that we sometimes see is the farmers, and this I think happens more often when the farmer is a little bit more experienced. So they recognize that every time they recognize patterns in their disease outbreaks. So they'll say, oh yeah, every time I move in fish out, a week later they have high mortality, so I'm just going to load them up with antibiotics because I think it's a bacterial problem. So those types of uses of antibiotics are overuse and can be prevented. And then the last one I want to talk about is improper use or improper dosing, and that's usually an underdosing that we see. And this one's actually harder to address. Underdosing of antibiotics has been demonstrated to drive AMR, and what it does is it will select for bacteria that can upregulate certain genes or downregulate certain pathways that make them more able to cope with antibiotics. So over time, you end up with AMR. So it's not as intuitive as just overuse of the product. But I have here, it happens, so underdosing happens in, I think, almost all aquaculture industries that I've worked in. I have here data from the salmon industry, so a very sophisticated industry that has a lot of measures in place to properly use antibiotics. And you can still see from these data, this is thousands of fish taken over 3 years and many different farms. So we see that the two products that are commonly used in aquaculture, fluoromphenicol and oxytetracycline, we see with fluoromphenicol a product that's very hard to use in warm water because it has a very short half-life. So 50% of the fish that where they measured the antibiotic tissue concentration in the fish at the end of their final day of treatment. So they should have been over the MIC, which is the minimum inhibitory concentration for that particular bacteria. They were dosing them for this. They should have been over that. But 50% of the fish they tested were below that level, underdosed. Again, oxytetracyclines, a lot less under the threshold, but because that's an easier drug to give in, it's longer, has a longer half-life, so it's easier to dose, especially in warm water. But still, we have 10% of the population underdosed. In an industry that is very, they pay particular attention to production. So with small-scale farmers, we have, and part of the reason we have this underdosing is that we're mass treating a huge population. We're not pilling every animal, so they eat differently. Some of them are sick. They might not get the dose as much, this kind of thing. But we sometimes have poor biomass estimations, so we're not even giving the right amount to the whole population. We have improper delivery of medicated feeds. We have poor quality drugs, and we have environmental leaching. And I want to spend a little bit of time on these problems when it comes to in Hong Kong, or I think for most small-scale farmers. So biomass estimations, a lot of our farmers don't have an accurate number of fish in their ponds. So they keep adding fish as they sell them out, but they don't have perfect records. And I joke around sometimes, and I'm like, is this for tax reasons? Like, why wouldn't you know how many fish you've got in there? But it's actually, I think, just the way they've done it always. So, I don't know. They have mixed species in their ponds, and they eat at different feeding rates. So when you're medicating, you need to know the feeding rate. And they sometimes eat different foods. They have a wide variation in size because, again, sometimes they just keep the pond going. they don't fallow it. So you might have different year classes even in there. So it makes medicating the ponds the ponds a bit difficult. The other thing that we've observed is fraudulent products on the market. So when we first started, we wanted a small pharmacy, and so we tested products that we bought. And turns out not all the companies that sell medication have the same standards as those Swiss companies. So the quality is sometimes not always great. So I'm giving you some numbers from a paper we published. We tested 15 products, and about 9 of them had at least 10% less than what the label said. And some of them had nothing. No, I don't know what they were. They don't cause AMR if it's not an antibiotic, but still, this is a problem when we go to give medication if we don't know the active ingredient in that premix that we buy. And then the other problem that we have to deal with is hand mixing of medicated feeds. So in Hong Kong, and I think in many countries in Asia, there are no commercial feed mills that will make medicated feed. The salmon industry actually has medicated feeds made in the mill. So the mill has better equipment, and they have a better way of putting that medication in the feed so it doesn't leach out as much. We have to top coat if we're going to use this. So you can imagine that there are some leaching issues depending on a number of factors. But we tested this in our lab because we were recommending some of these things. What we found was so we used oxytetracycline, OTC. We used it at a concentration that we would normally use for a 1% feed rate. And after about 5 minutes, we let it soak in the water, and then we checked the water. And we had after 5 minutes, about 0.3% of the antibiotic was off the feed in the water. Maybe a bit more, but that's how much we were able to detect in the water. Not too bad, but 2 hours after, we had tenfold more, so 3%. So the problems there is you have antibiotics going into your environment, and that 2 hour isn't that for bottom feeders. Actually, you might have a 2 hour wait before they feed. So you're getting antibiotic at a low dose in your environment. And remember, for us anyways, the bacterial infections we see are usually the Aeromonas or the Vibrios, things that are already in your ponds or in the environment. So putting antibiotic at a low dose in the aquatic ecosystem could be problematic for you. And it also means you're underdosing. So you're likely not going to achieve the goal that you had in the first place, which was to treat the animals. So the consequence of underdosing the fish, as I mentioned earlier, it does put a selection pressure for AMR. And the other thing it does that a lot of people don't really think about a lot, it's taken me a few years to consider this, but if you're not feeding if not all the fish are getting dosed properly, some of them are going to maybe retain the bacterial infection, especially if it's a more chronic infection. So once you're done with your treatment, then those fish could serve as a source of infection for exposure to those fish that are now, they were treated successfully, you had a nice dose in them, but now they're metabolizing that drug. So they are now in this zone of low dose subtherapeutic levels of antibiotics. So again, you get a double whammy on that low dose treatment. So what are we doing to address? So these are the things that it's complicated to try to address antibiotic use in aquaculture, because there's a lot of issues that can actually lead to this state. So what did we do at City University of Hong Kong, which is where they have a new vet school. So we needed a vet service to help train our veterinarians. So we ended up contacting the local government and pairing up with them to provide a subsidized veterinary service. And what this service does is farmers don't have to pay. They can call us, and we respond within 24 to 48 hours, and we try to figure out why their fish are dying. So we built a diagnostic lab with capabilities of dealing with fish aquatic systems, so the bacteriology, the PCRs for the virology, and we help the farmer deal with the problem at the time. If they have a bacterial problem, then, and we have a set of criteria. So we don't just hand out antibiotics, but if they have a bacterial infection, if they have a good estimate of their biomass, if they're feeding pelleted feed, if they're feeding, if the fish aren't close to market, then they're a good candidate to use antibiotics. So at that point in time, we offer them that option. We have antibiotics in our pharmacy that we quality assure. So anything we buy, we do HPLC on, so make sure that we know what the concentration is in the premix, because you buy antibiotics in premix to put in feed. And we don't make a profit. So there is no incentive to sell drugs. We sell at cost. So that's actually really important. If there's a service that makes money selling drugs, then there's a conflict of interest. And in fact, we even, after 2 years of running this service, we noticed that people might have been, our drugs aren't super cheap because we buy good drugs and we HPLC them and we can't lose money, so it's no profit, but it's still higher than Taobao. So I think people were getting a diagnosis from us and then sometimes going to Taobao to shop. So we talked to the government and we convinced them, and this was no small feat, to subsidize the drugs so that we were competitive with Taobao. So we said, look, we don't want to give drugs away, and we don't. Like, they monitor how much we give out, and we really don't give it out unless it meets our criteria. But we were able to convince them to pay for half of the price of our nonprofit drugs. And since then, I believe that farmers don't go on Taobao to buy drugs. Like, I'm pretty sure that our clients, we've also got a good relationship with our clients because we're free and we sometimes help them. At least we try all the time. But and so that has actually, I think, reduced some of the illegal use of drugs in fish farming. I don't think it was that much, to be honest. But we also, the other thing that really is important here is that if you go on a site and there's a problem and you solve their problem, then you have an opportunity to target prevention strategies at that time when it's still fresh in their mind that if I did this and this caused this, okay, why don't we think about what you can do to stop that in the future? And it's very strong messaging. Instead of saying, come to a workshop in 6 months' time when you've forgotten that you actually have this problem. And I'll even say that it takes usually 2 site visits, maybe 3 site visits for the same problem before they listen to the prevention strategy that might be effective. So it's not a for sure thing they're going to do it on the first go. And then the other thing that we have money for from the government is to conduct workshops. So the times when we want to do like big industry messaging, then we'll run a workshop. We do good practice workshops. We'll sometimes ask them what they want, and we'll bring in experts if we don't have the expertise in-house. And we'll do translations, because all our farmers are Cantonese speaking. Very few speak English, so we have to translate. We'll also, like we've done, some of the first workshops we did were on withdrawal periods to make sure that they understood why we need those and how important they are, how to mix and deliver medicated feed so they don't just dump it all in one go and with no coating at all on them and things like that. So these workshops have been quite popular. We also have a WhatsApp. All our farmers are on WhatsApp. So when we have a problem, we actually send out a group message to all our clients. Like when it's really hot and there hasn't been a storm yet, but the storm is coming, we're like, remember, this is the season for turnover. Make sure you run your aerators in the middle of the day, even if you don't think you need them. So we'll message them certain, we don't abuse it, but we try to message when we think there's an issue in the industry. All right, So I'm going to finish with a summary of our vet service. So in 2018, I was alone. I had 8 clients to start with that year. And I didn't do a lot of cases, but I got it going. And I just want to point out that 24% of my cases were bacterial and required antibiotics for resolution. Now, this year, we're already at 100 cases, and it's the middle of the year. So we've rebounded from COVID. We're getting more and more cases, and farmers. We have 80 clients now, I think. And we also do preventative. We follow up, and we do preventative calls when we have spare time. So we do about 300 last year, almost 300 site visits. Now it's going to be over that for sure. So we make sure we have a presence in the industry. Those workshops, we do about 2 a year, sometimes more, but 2 a year. And then the last thing I want to point out about this vet service is the government also gave us a little bit of a slush fund to address problems that we identify in the industry that need a little bit more research. So like sometimes we have minor species that they're farming, and we don't know about, if we don't want to use any antibiotics in them because we have no idea what the withdrawal periods are. So we can actually have a bit of money. We have money set aside to test those things. So we also had money to look at probiotics for the farmers because they wanted us to. So we respond to their needs, and if we see that there's something that's food safety related, they might not identify it, but we identify it, then we'll work on that kind of topic. And so we've looked at how to clean water because the water is a problem, these kinds of things. We've actually even looked at top coating feed with other materials to reduce leaching. So all of this is working towards that misuse of antibiotics, the improper dosing, the overuse, and the inappropriate use. So. And then the last thing I want to share with you is anytime we find a bacteria, we do a sensitivity analysis. And so a couple years ago, we decided to look at our trends over time. And it was very interesting, and we actually published on it because it was surprising even to us. So I want you to pay attention first to the first 3. These are drugs, antibiotics, and the first 3 solid lines are antibiotics that are commonly, they're labeled in other countries. They're formulated for fish feed, so you can buy them in a premix, and those are the ones that usually are used in aquaculture. And for our service, we decided we're not going to use the baby blue one, which is an oxalic acid, a quinolone, because quinolones are quite important for human use. So we were trying to reduce the use of those. And when I first started, I was, coming from Canada, there's hardly any antimicrobial resistance there compared to this kind of stuff. So 90% of our isolates, it wasn't a lot of isolates, but still had quinolone resistance. Also these other 2, but and then we stopped. We didn't use it, and because we were helping our farmers with the medications that we had, I don't think they used quinolones, and we saw a huge decrease. And it's now, very very little. The first year, I decided I wanted to just use chloramphenicol because chloramphenicol is not a human drug. So I thought, we'll just use chloramphenicol, and then. But chloramphenicol, recall, when I mentioned it's very hard to use in warm water because it's such a short half-life, so you can just never get it to the therapeutic level for long enough to treat the fish. So we were getting poor response, and I think actually I might have caused this. I'll own that one. But then we stopped using it, and we went to the oxytetracycline. But even if we just use oxytetracycline, you can see we're really quite low now for AMR. And okay, it's N of 1, so, But what we tried to do here is say, okay, maybe, all of the drugs were going down. So that's why I have included here 3 other drugs that are, they're a little bit more price sensitive, at least some of them, and they're also not in a formula that's easy for the farmers to use. So they tend not to be, I don't think they're using these commonly, in Hong Kong anyways. So we wanted to see what the pattern was with that, and you can see it just doesn't waver at all. We might have a little problem in Hong Kong waters with these drugs, but I don't think it's from aquaculture. So the other thing we thought, well, maybe they've stopped using these products, these three that were kind of important to us in the human medicine. So we went and looking at community-based antibiotic use is quite hard. It's not recorded very well, but hospital-based antibiotic use is. And for Hong Kong, it's actually publicly available. So we looked over the trends over the years, and actually the usage of these products is all going up in Hong Kong. So we don't think that our drop was because of a reduction in human use, which, you know, we have millions of people and we don't have that many fish, so that might have been a reason. So anyways, that was a very encouraging, at least we weren't making it worse, and we might even be making it better. So can access to diagnostics and veterinary services reduce AMR? This data suggests that maybe we're having a positive impact, and it makes some sense if you provide farmers with the reason why the fish are dying and a solution for them, then maybe they won't go and use antibiotics on them. So it does resonate with biology. And we've now started, we've helped Singapore establish a vet service for their fish farming community, and we've just put in a grant for USAID to see if we can't get a pilot project there to see if that helps AMR. So hopefully we get more than one location to assess this. The real problem is who pays for it, because it is quite costly. And for sure, Hong Kong, they've gotten the Cadillac. You know, they've given us money for research and so they're very generous. But not all socialized fish medicine, I think, can exist in a lot of places because of the cost. So I will finish there. Hopefully I didn't go over too much because I'm the moderator. And again, the AFCD government in Hong Kong sponsored all of this. So I think we have time for one question from the audience. And maybe some of the questions can be delayed till after, but yep. Well, they do not, but we, as the VET program, could bring them into the country under registration. Like we could write a prescription for them if we could. The problem is that they have very small numbers, and they import fish. We don't have a lot of hatcheries, so they're already importing fish that they bring in the disease with them. So it's quite hard to, like the viruses, it's quite hard for them to vaccinate, and they have such small numbers that it is really hard for them to use. We've discussed that a bit with them, but there's only a few of the farmers that are big enough that would even consider it. Okay, sorry, I'm the moderator. I have to introduce the next person. One moment. Let me get my cheat sheet. All right, sorry about that. So our next speaker is Dr. David Verner Jeffreys, and he's now with WorldFish, but he used to be with CEFAS when I knew him. He's going to talk about building capacity to tackle the threat of antimicrobial resistance in aquaculture in Asia and Africa, using a One Health approach.
Next slide up. That's down and up. Yes. Good afternoon, everybody, and thank you very much for the opportunity to come and talk to you. As Sophie's alluded, I've recently moved from CEFAS, Centre of Environment, Fish, Aquaculture Science, which was there for a very long time, before joining WorldFish, helping take on some of the responsibilities of one of our next speaker, Mohan, which I think everyone will know a lot better than I do. So my talk, not surprisingly, is going to be quite a lot of my old CEFAS life, but maybe sprinkling a little bit about some of the activities that WorldFish have been doing in this area as well. Fortunately, I did do some of this work together with WorldFish, so there's a sort of some integration work. I quite like this slide. This is an unnamed Asian country, and what we can see here is maybe illustrating the One Health principles. We have a layer unit, a layer unit, and then we have an outflow pipe. Then we have a little pond, around that pond we have vegetables, and in the pond are fish. So what we're going to be talking about a bit today is putting things within a One Health context, particularly around the transfer of AMR and how we might manage it. Thank you. So starting out, following on from Sophie said, very good, thank you Sophie for laying the scene here so well. In fact, the antimicrobials are used to control bacterial disease in farm fish, everyone's aware of that. Vaccines, maybe something to talk about later in the discussion is interventions. We know that they can be very effective, and certainly in Norway, in the salmon industry, they're highly effective and have significantly reduced use. A general aspect, and we'll talk about challenges in Asia and Africa in particular, is more widely, there's actually access to antibiotics as a general issue for the industry, certainly in terms of regulated products that can be used. AMR in aquaculture. So resistance in aquaculture, antibiotics, is not new. There's a Japanese researcher, Aoki, in 1992, that when he looked at bacterial pathogens, showed already resistance plasmids were present in nearly all drug-resistant fish pathogens. So you think that antibiotic use in agriculture and fish farming probably coincided more or less with the use in humans medicine. And sure as night follows day, people using antibiotics in aquaculture, you saw the signal within the organisms that they were developing resistance. And before we get into the meat about what people should do and whatever about it, there's a lot of actually, sometimes we almost have to take a step back. There's also the evidence that this is a real issue. I mean, when we think about resistance, really, the most important thing really is, frankly, the clinic and those things. They are important tools within disease control within animals. But what is the real evidence of transfer? Because we always talk about One Health, and we're going to talk a lot about it. But what actually evidence of this takes place? And I think there are certainly some signals from molecular data within fish pathogens showing that highly similar resistance genes, and particularly the flanking elements that these elements sit on, are highly homologous. So that shows there is movement of these resistance genes through space and time. And it's probably sensible to suggest that under selection pressure antibiotic use, these will be fixed. And I think there is evidence of that. A nice paper, is getting on a bit now, from Tim Welsh from the USDA. And he did a really nice piece of work showing that an important class of plasmids, and these are, for those who are not microbiologists, sorry, I'm going to occasionally lapse into talking about microbiology and stuff. I had to put a few little extra pictures of things for those who are not microbiologists. Plasmids are mobile elements that can move between bacteria of different species and different genera. So when we talk about the One Health approach, we know that resistance genes can be mobilized on plasmids, and they can move between bacteria of different species and genera. So the type of resistance that can cause a problem in a fish pathogen can cause a problem in a human pathogen. Here we have a great one, because these NKC plasmids can cause plague. You know, you don't get much worse than that. But also we've seen similar plasmids in a variety of other pathogens, including in fish and also in other cattle pathogens and the like. So there's some evidence that these things move around. You can argue about what really forces that and how it might be selection, but maybe a bit of background saying this stuff does happen, and we probably do need to be, this is one of the reasons I think we're putting so much effort into kind of controlling and managing it. Sorry. Okay, so. Classic sort of figure, one that we did, but there's lots of other ones around as well, showing the theoretical transfer of AMR bacteria and resistant elements between different compartments. I think everyone recognizes this takes place. There's some debate, frankly, about the strength of the arrows. What are the really important arrows? They're probably more, frankly, human, but we know it does take place between some of the other compartments. And particularly in our relevance to this audience today, the aquaculture. Water is a great way of moving bacteria and resistant genes between different compartments. What should we do? Well, I haven't put a slide up here, but I think everyone's aware that the quadripod, David's going to talk a little bit more about One Health and all the rest of that sort of stuff. And I'm not going to bother, because you will hear more about operationalizing that. But certainly there is a need to use a One Health approach. Countries now have national action plans for AMR, and out of that is flowing guidance and guidelines. And there's a couple of important guidances, which I think everyone here should be aware of, if you're not already. The Aquatic Animal Code, from WOA. That does lay out, probably more at a country level, about how you might want to put away AMR surveillance programs. But a really nice piece of work that's come out from this region, FAO and Parks, is a set of monitoring guidelines for antibiotic resistance and residues. Anyone's interested in that really should look at that. It's a really nice piece of work. Involved a lot of collaboration, even CEFAS tangentially, but so Mohan and others. A lot of people fed into this, and this is a great resource. I really recently shared it with people in Africa as well of similar challenges. And I think it's a good place to start. These things are complicated. They've concentrated more on the pathogens of fish rather than the commensals, but it doesn't matter. A lot of principles, there's a lot of guidance about how to take your sample and do your analysis. So I would suggest we're going to talk a little bit, frankly, a series of anecdotal case studies about how you might fit within that framework. I think you need to think about a holistic. You have a big framework, everyone needs to be involved, and we're all doing a little bit. And think when you rest of my talk, that's what I feel, and when I looked at it afterwards, actually, we're doing bits of this, but you need to be within a coherent whole. And I think the WOA and the FAO guidelines are good places to kind of start to go to. Capacity, what should we do? Capacity building in Africa and Asia. I think, as Sophie's alluded already, we know that the systems in many countries are weak when it comes to the regulation for the control, application, diagnostics of the diseases. All of that needs work. And there's a need for different elements of strengthening. And again, I think using a collaborative approach, strengthening regulations, strengthening AMU surveillance systems, collect data on use, analyse and reporting, both to farmers, but also to national authorities and ultimately to WOA and others. AMR surveillance, a number of different aspects of that, and quite a lot of what I've been doing the last 5 years is actually doing bits of this, and I'm going to illustrate some examples. Improving diagnostics, following from Sophie, absolutely. When you go to countries, you can't do an antibiotic susceptibility test if you can't culture bacteria, identify it. You need that basic capacity in place. Coming back to that is if you're going to look at how you can look at lots of different things, you probably need to focus on a few particular drug-bug combinations, and the Europeans, Americans, and others spend a lot of time trying to refine target drug-bug combinations if you're going to do a surveillance program. I like the frame surveillance is not monitoring here, we're talking about informing action here. Laboratory strengthening, I'm going to talk a little bit about that, the nuts and bolts about how you might do antibiotic testing. Quality control, proficiency testing schemes. And my old colleagues CEFAS and DTU and others, they have really nice schemes where they'll send isolates around to laboratories to engage in ring trials. So actually you can have some confidence that what you're reporting as resistant is really that or not. Workshops, sharing information, best practice. We all do a lot of that. I'll give some examples. But all around humans, it's all around the people. It's all very good having the equipment, the fancy MALDI-TOFs or the next generation sequencing, but you need the human capacity. Training, mentorship, we'll talk a little bit about that. Building communities of practice, sharing what we have to do. And then I think maybe this afternoon we're going to have a bit of a talk about this, about the practical microbiology in an aqua context. It'd be nice to talk a little bit about interventions. Quite a lot of what I've been involved with, one of the reasons I've moved to WorldFish is to be able to focus a little bit on the other end of it. It's always, surveillance is great, generating data on what's going on, but what are we going to do about it in terms of interventions? Biosecurity, vaccination, other control measures. Strengthening national aquatic animal health systems. I think many people in the room has informed governments to help build better, more practical. I like to use the practical. Sometimes when you name, like for instance, I'm not going to name the country. One country has at the moment a regulation that says you shouldn't put antibiotics in feed for treating fish. Well, you straight away cause effectively a grey area of regulatory practice because everybody knows who's in the field, Sophie's a fish vet, you know, that's the only way you're going to get your antibiotics into fish effectively. So straight away you're kind of perverted in terms of the social side of that. So you need effective regulations that are actually going to be used and applied properly. Because as soon as you kind of step away from doing what you're told, then maybe you're on the slippery slope to other questionable practices. Just a thought anyway. Promote and embed the One Health approach for sure. Strengthen early disease detection and all these other things. And collaboration with WOA, FAO, there's lots of support out there, and I think Melba's going to be talking later online. And provide training, okay. My last job with CEFAS was I was part of this great entity, the UK FAO Reference Centre for Antimicrobial Resistance. The FAO have established a number of reference centres. One is broadly more terrestrial veterinary, although CEFAS was part of this, and there's another network, which I think Melba will talk a little about later. And this was collaboration between the Veterinary Medicine Directorate, the Animal Plant Health and CEFAS, all agencies, Department of Environmental Affairs. So basically, you find a one-stop One Health solutions to AMR support. And we got funding from the Fleming Fund. I think everybody here know what the Fleming Fund is. This is a UK aid program to support the development and implementation of AMR surveillance within eligible countries. Lots of other activities you did, but looked upon in that framework. Here's a good example, working in Bangladesh with WorldFish colleagues, actually, and the FAO ECTAD team there with, and we had, we brought people together, the Bangladesh governments, to look at identifying gaps and how we might improve AMR surveillance. What was really nice was bringing people from the terrestrial side and the fish side in Bangladesh. Now, we've got Bangladeshi people here. Apparently, trying to get terrestrial vets, aquatic vets, or fisheries people, even in the same room to talk about shared issues was unusual. So we managed to do this. So AMR, in its perverse way, sort of actually brought people together who wouldn't normally talk. And those conversations were really, really fruitful. So that participatory rate, bringing together the different aspects of government and other stakeholders. And from out of that, we identified areas where we can improve. And we found actually it was interesting, when you looked at it, you found the terrestrial sector was stronger for some aspects of this, and the aquatic sector was stronger for others. And actually, it was a real complementarity that came out of that. Laboratory training and testing, a lot with my old team. We used to work, and actually the other thing was working with our Applied Health Agency. It was really good working with the terrestrial veterinary microbiologists and us, because we actually soon realised we speak much the same language, and we'd go out together and work with the competent authorities from the fisheries and the animal health thing to develop and improve's the right word? Yes, it is. I don't see why not. Actually look at their processes and systems to actually help them undertake their testing in a more rigorous way, using the appropriate methods and looking about such things as laboratory safety, all the way through to producing their results and reporting them. And also did some nice research together as well. Another piece of work that I did with CEFAS with colleagues from WorldFish, actually towards the end of COVID. Sabrina Hussain and Shafiq Rahman from the health team there did a really nice piece of work of developing a wet market surveillance project. I think, they're little snapshots of things you can do, but I think you need to see these as within the broader framework and feed this information in to develop surveillance programs. I'm going to talk a little bit about fundamental microbiology here. There's a lot of, how do I put it, a lot of the papers out there at the moment are pretty free with the use of the language and terminology compared to, say, the terrestrial structure. Now, there's someone I've had the privilege of working for a very long time now, Pete Smith from University of Galway. Anyone's in the AMR world in aquaculture will know Pete. Still very active, still driving us for rigor in what we do and how we do it. And he's pointed out, how can we share data about resistance or susceptibility if we're not using the same methods and using the same reporting criteria? This is probably a nice piece in a journal I work with, Disease of Aquatic Organism. We called it 8 Rules for Interpreting the Quality of Papers. So it's basically, if you're doing this sort of work, go through this and try and use this fairly rigorous framework when you're developing your programs. And I won't go into the steps here, but basically it's using standardized approaches. Be careful about using resistant. There's all sorts of issues to do that, and you talked about that. So it's basically, here's a good sort of kind of guidance about how to go about doing things. One of the key challenges we have in aquaculture pathogens is if you're a clinical microbiologist and you use the term resistant, you know that's probably based on clinical resistance, that your susceptibility has clinical relevance. You know, you're a doctor, you're medicating your patient, and you know that it's unlikely to work because its susceptibility is the amount of antibiotics it grows in is high. It can grow in higher concentrations than is likely to be achieved in plasma. Coming back to Sophie's things. In aquaculture, we're limited. We don't have clinical breakpoints for most of the pathogens. For one of the things, it's really hard to set them. These are temperature sensitive. We're working with animals and pathogens that the temperatures are varying on things. All sorts of reasons. What we tend to favour now is what we call ecological breakpoints. And I'm just going to try and just briefly summarise what I mean by this. Sorry, I've lost the slide. I did have a slide of because I know a lot of microorganisms in there, which has a picture of a disc diffusion plate. Now, most of you have probably seen them. It's a disc and you'll see a little disc with antibiotic in there, and you'll have a zone of clearing around the disc. And you measure that, and what you tend to find is if an organism is less susceptible, that zone of clearing reduces. So when we have here is these are zone sizes. So basically, the bigger the zone size, the more susceptible the pathogen. What you can do is if you have a plot organisms that are unlikely to have any intrinsic resistance, you can set an ecological break-off point, cut-off point. So basically, you can say this point here, if your organism its zone size is less than that, it's showing what we term non-wild type susceptibility. Okay. And the same goes for if you're growing. The other main method we use is, as well as putting a disc on a plate and seeing the zone of clearing, the other way is on a plate. Typically, you can put lots of concentrations of antibiotic and see the minimum concentration that the organism will grow in, and from that you can set the minimum inhibitory concentration. And again, what you can use this data to set interpretive criteria. So we can all use the same language and say that this is non-wild type. And one of the things that I'm still doing, hopefully, and working with colleagues around the region as well, and parks and others are involved, and internationally, is developing, setting provisional cutoff values for these organisms. And here's an example of the sort of piece of work that we're doing. I'll just go back up again. I'll say that. So for setting interpretive criteria, what we're looking to do, and we have a special issue in DAO at the moment, which we're trying to bring this data together, is we want labs to come together to each measure, using appropriate methods, the susceptibility to different antibiotics. And then what we do is we pool all our data, and then we can use that to set our ECOF or ECV values. And there's been a really nice sort of flurry of activity here, and we're starting to have quite a big range of bacterial key pathogens, things like Vibrio angularum and others very relevant to the region, so that we can go, yes, we've got working with the same language there. You can use the term non-wild type, wild type. So this is something that we're sort of driving in terms of standardization, data quality reporting, and harmonization. So that's been a really, really good thing to have been involved with, I think. And it's a lot of laboratories involved have been in. USDA has been heavily involved. Melba and others have been supportive. So yeah, we've had strong support from a number of the organizations for this. So here's an example of the type of data you might have from MIC values. So here's an MIC, micrograms per mil of different antibiotics, and you can see what we do is there's about 170 isolates that you've tested, and from there you can plot their distributions. And if what they call non-wild, and then you can then work out from that if it's less than or equal to 1 mg, it's likely to be non-wild type. Okay. So if it's coming in greater than everything. So that's how you set. That's basically a quick intro into how to set MICs and how we bring labs together to generate data. Capacity building. It's all about the people. One scheme that I've really enjoyed working with is the Fleming Fund have what they call a mentorship fellowship scheme. And the idea is what you do. The authorities within the different countries that are eligible to the scheme, they select typically mid-career employees, government scientists, and others, depending on the specialism they're trying to develop a fellowship in. And they don't leave employment. They're not master's students or anything like that. They're still doing a job of work for the government. And typically, like here's an example of two in a minute. And what they do is they get a year or more of support from a lead organisation, an external organisation. In this case, when we were at CEFAS, and so for instance, here's how this scheme works. There were two fellows I was involved with. There was Eric from Fish Health Unit of the Fisheries Commission, and Dr. Sulia Adeleke, a Nigerian agricultural AMR fellow from the Department of Veterinary Pest Control in Nigeria. And basically, we worked with them over a year, gave them a lot of experience, and apparently they said they benefited from a lot. And the idea is they should then go back as ambassadors for AMR within their countries. And there was a variety of mechanisms by which we engaged with them, gave them training, they met the mentors, and they came in person, we supported them going to conferences and the like, building that capacity to act as AMR ambassadors and have the right technical capacity to support what they were trying to do. They also did some work. We helped them with some surveys in Ghana and Nigeria, and rather reporting what Sophie said, and again, probably of no real surprise to anybody in the audience. Whoops, where's it gone? You can see that most application of antibiotics was not under professional supervision. And a lot of them were using antibiotics that were probably inappropriate. So there's, again, evidence of inappropriate use. And there's some pictures of them. Biosecurity, I haven't got my time for interventions. We've done quite a lot of work with WorldFish, of working particularly in Bangladesh, to develop practical biosecurity guidance and guidelines. And here's examples of some workshops that Athena, my old colleague, and Sabrina, my present colleague, working together to develop guidance and guidelines in Bangladesh. Vaccination, other control strategies. I think there's a real problem. We really need to talk about that. What are the barriers to vaccine uptake? Maybe as part of the discussions is because we know they're great tools. Why aren't they being used more widely? And there's lots of reasons for that. And I think maybe it's useful as part of this workshop to kind of explore some of those. We also do research in terms of antibiotic susceptibility testing of organisms, and this is some work we did actually in the Gulf states looking at E. coli, and we found, frankly, very high levels of resistant E. coli and a lot of carriage of a lot of concerning resistance genes. What are we doing next? Rather than with a closeout slide, I think everyone recognises you should be employing a One Health approach. We need to be working together, and I know I've just joined WorldFish. This is stuff that's been set up by Mohan and Jerome and colleagues. We are hoping to work quite a lot with the Fleming Fund. Not big programs of support, but there's the regional grants in Asia and Africa. We'll be supporting them. The Fleming Fund fellowships. So hopefully, I'll be mentoring a couple of fellows from Nigeria again, and also recently completed. We supported. While I was with CEFAS, actually helped run it. We had a regional AMR aquaculture workshop. So I think, in summary, there's a lot needs to be done. I think we all need to work together. One Health is my way as an operational tool, is using a collective approach to look at all the aspects when you're trying to solve the problem. And I think with that, I'll thank everybody. I've got a long list because a lot of old colleagues I work with, a lot of new colleagues I work, but have all contributed to some of what I've been trying to talk about today. But thank you.
Thank you, David. We have time for one question from the audience. I think the questions online will be sent to you.
Good.
Yes. Yeah. Where's our little runner?
Thanks. Hello. Thanks for the excellent presentation. And this is to Karen. You both, I don't know, make sense or not. It's from aquaculture perspective for small scale farmers. Somebody said that, okay, there are many reasons for disease to cultivate in small scale farmers in their little pond. What is the major reason and what is the preventive measure and simple message to them? Is it connected to stocking density and feeding practices, or is there anything like in a simple message which can be considered as a preventive?
I will start. I'll let Sophie as the vet follow. But yes, in our experience, typically it's poor management. Lack of oxygenation, overstocking, these sort of issues. But also poor biosecurity as well is, and by that often, when you actually dig deep into what farmers are doing on some of these small farms, you find that and you get into the social economic side, is the reasons behind it. There's good reasons behind it. It's not irrational. It never is irrational. They're continually bringing seed in. And they're often, micro-cropping as well. These sort of practice are you're kind of likely to be increasing risk. So those are the reasons. Some of those things to actually deal with are quite hard, because this is not irrational. It's not irrational. A farmer is taking continuously cropping, because that's the way they're paying off their creditors. But the consequences of that in biosecurity practices can be difficult. So that whole aspect of, actually how do we implement context-appropriate and culturally appropriate biosecurity is something I don't think we've always been, particularly, from the West, haven't always had that lens, And I think going, you should just do that. And actually, you need to be a bit more aware about what's going on the ground. So, but maybe, I don't know if Sophie wants to follow on that. Sorry, throw that back at you.
I agree. So for our farmers, a lot of times it starts with that environmental problem, and it's because maybe they haven't fallowed their pond in 7 years or more. And so You get the water quality issue. Maybe they're overfeeding when it's super hot, then they get an ammonia problem. So it's management, but because they're not monitoring as well as some of the bigger farmers, they might not pick up on it right away. And this continuous stocking, their markets are live markets, so they're not doing all in, all out because they don't have a market for that. They might not have a processing plant that'll take the fish, so they have to just small sales every day. So there's a lot of scenarios. I mean, sometimes they'll bring something in, of course, but oftentimes it's more of an environmental problem that deteriorates, and then they. And I don't know if you noticed, but our data from our cases, we've shifted from bacterial problems over the last 3 years to environmental problems. That's why I'm like, oh, we're picking things up earlier, because when we go out to the farms, we're like, new clients, they'll call us after 50% of their farm is dead. And I'm like, I can't help you that much, but okay, let's try. Next time, call me earlier. And if I do that a couple of times, then they start to call earlier. That's what we're seeing in the last 3 years. A lot more environmental problems, a lot less bacterial problems. So anyway.
Thanks.
Okay,
Thanks, Sophie.
I want to keep on track.
That's
so next
Thanks.
speaker, thanks, David. The next speaker is also a David, Professor David Bass. He is from CEFAS, still from CEFAS. And he's going to talk about systems approaches to in aquaculture for animal and pond health management. And maybe make sure you speak up to the mic, David.
Okay, brilliant. Thanks very much. Just stick your hand up if you can't hear me. I hope everyone's still awake after that nice lunch, but I'll be watching for nodding heads. My talk's in two parts. The first bit is on systems thinking and linking that to One Health and some suggestions of ways forward. And the next bit is still about systems, really, but looking at microbes and pathogens in pond systems and pond animals, and how we can take that kind of approach to better management and understanding of those systems. Yeah, it's working good. So, systems thinking, there's a lot of people are talking about this at the moment. There's a lot of vagueness about it. Critics will say, isn't it just talking to each other? We do that already. Well, no, it's more structured than that. But, of course, a lot of disciplines are inherently systems. Aquaculture is one of them in its many forms. Ecosystems, the clue's in the name, is absolutely a systems thing, and you can only understand it by taking systems approaches. So maybe we're already a few steps ahead, but actually some of the tools and frameworks around systems approaches are quite helpful. So systems, here's some explanations of what systems are. They can't be understood by looking at their individual elements. So you need to look at the whole, greater than sum of the parts, and a collection of tools are available. And this is just an example of one of the graphics around those tools that I've found, where you ask questions of yourself about the problem. Do we understand the problem? Do we know what causes it? So we're already at a sort of solution stage, or are you still trying to understand what the problem is? Similarly, questions about the stakeholders, the policy setting, the ambition, and going through that process will help you think, yeah, this really is a systems approach, so I need to work out what the available tools are for working within that systems framework. I'm not going to go into that today. There's loads of stuff online. Look it up. It's actually really interesting. I was looking for papers that explicitly have been using systems approaches in aquaculture, just to illustrate the point here. And I didn't actually find that many. As I say, there's a lot where systems approaches have been used generally without stating it, but sort of explicit approaches, I haven't seen that many. Here's one of them, looking at interventions to reduce antibiotic use. And tilapia farming in Egypt involves people in this meeting, so this will be familiar to some of you. But just going through these 6 steps here, this is the sort of graphical abstract, I think, of the paper, of the process that they went through, taking into account the different areas that are of stakeholders and of relevance, so human behavior, pathogens and host, environment and governance. So if you think about all of those questions and address all of those points in that broad context, then you can start to draw a systems map. We'll see an example of one of those in a minute. This is one of the other papers, again, involving quite a few people in this meeting, this time looking at hotspots for antibiotic resistance, emergence and selection, and where humans get exposed to it as part of the aquaculture system. So they looked at these human exposure points, you can see in in this thing here, and they looked at where that could happen in terms of consumption, the occupational efforts of the people involved, and environmental aspects, and then they came up with this map here. And obviously we're not going to go through this in major detail, but I've just illustrated a couple of the symbols here that they use to indicate hotspots for AMR emergence and hotspots for human exposure to AMR. But you can see the value of this kind of map and the fact that it can be applied to many different systems and tells you a lot more about how those hotspots emerge and their relative importance than just listing them on a piece of paper. So this is, I think, a very good structure for addressing lots of the issues that we have in aquaculture and the things surrounding it. So the One Health is inherently a systems thing. We don't call it systems necessarily, but it is exactly that. And there's a lot of vagueness around what One Health is as well. So this is a slide just to summarize certainly our take at CEFAS on it from a paper that we published a few years ago in Nature Food up here. So the way it works is we've got this One Health wheel, as we call it. And the important point is that One Health is an integrated approach that balances and optimizes the health of human, animals, plants, and ecosystems because they are so interdependent. You can't look at them in isolation. There needs to be a strong research, evidence, policy, and legislative basis for it. And then it can be measured by these 15 success metrics that you can see listed around the outside of the wheel. So that interlinkedness is the key here. But how do we operationalize it? I mean, as David said, it's one thing sort of saying these things and having these concepts and models, but unless you can work with them, they're of limited use. So one way that you can operationalize One Health as a system is this is another paper we published sort of soon after that One Health summary paper that explains the seafood risk tool. And the way this works is to, up here in box number 1, you just define your aquaculture scenario. So it might be tilapia farming, it might be shrimp farming in a particular place under a set of particular conditions. It might be bivalve farming for raw consumption. And then you go through a process going around here where you consider a list of hazards, so chemical hazards, animal pathogen hazards, and human pathogens hazards, and you make a customized list of those hazards depending on the aquaculture scenario that you have defined in the first place. You then apply a seafood risk tool, which we'll see a bit about in a minute, which involves looking at a risk mitigation matrix. They're measures that you can apply to reduce those risks. And then you come out at the end of it with a better understanding of what those risks are and guidance about how to mitigate them so that you improve the One Health of the whole system. And then on the basis of that, you can devise policies, best practice, guidance, and so on. So taking this example a little bit further, just to illustrate it, here is that bivalve aquaculture scenario for an export market where these are going to be consumed raw, so uncooked. And as I say, you look at the hazard categories which are listed on the left there. So 6 chemical hazards, 5 animal pathogen hazards, and 3 human pathogen hazards. And you score them for that particular aquaculture scenario. And that's the ring around the oyster here, where the relative importance and potential impact of those risks is listed. You need to take into account across the supply chain, from early life right through to consumption, that the relative importance of those hazards is going to change. So in early and grow-out stage, the animal health hazards tend to be the most important in this particular scenario. And at the right-hand end, trade and consumption, the chemical hazards are highest. So you need to break it down in that way. But you've ranked your hazards. The next step is you look at this risk mitigation matrix. Now, I've just picked 4 hazards here as examples so you can see how it works. But each of the columns addresses each of those production stages that we just saw on the previous slide. And then the 3 colored boxes on the right are the result of applying those mitigations. And the mitigations are, for example, using the guidance in the OIE, now the WOA code, on-form biosecurity, best practice measures, whatever is relevant, available support to reduce those risks, you can refer to, you can apply those mitigation matrices, and then you can bring down the risk of the system. So the uncontrolled risks in the red box are the highest. You apply a single control, and as in control 1, and it goes down. You apply the right combination of controls, as in the control 2 box, and the overall risk matrix goes down even further, and that will lead to more favorable outcomes in that aquaculture scenario. So that's a very brief summary of that paper, but I think it's a nice way of operationalizing the One Health approach. Another sort of concept that we've just sort of come up with quite recently, working on at the moment, is this idea of observable and easy measured indicators for organism and environmental health management. So this sort of goes back to the conversation at the end after David's talk and several others today, which is farmers can go out, and aquaculture professionals can go out and look at ponds and make some sort of fairly easy observations, both in terms of organisms, the environment, and to some extent the microbiomes and the other organisms in the ponds. So for organisms, easy things to look at are animal behavior traits, the condition of the animal, the vigor, environmental parameters we all know about, microbiota parameters. We can look at water quality indicators, fecal indicators, for example. We can do screens for specific pathogens of concern. They're all relatively easy to measure. That doesn't necessarily mean that they're easy to do something about. So that's challenge number 1, is thinking about, if we can measure these things, what actually can the farmers and other professionals do to mitigate those problems? There's quite a few answers out there. Obviously, it's received a lot of attention over the years, but I don't feel that we've brought together that knowledge in a way that can be distributed across the aquaculture communities, across countries, in a way that they can trust and implement some of those things. So I think there's a job to be done to really help understand what some of these easily observable indicators actually indicate, and what action can be taken on the base of that. So I think that's a collective challenge that we have here. And then you sort of go across to the difficult side on the right-hand side, and there are things that you can do, but I can't think small-scale farmers are going to be measuring the transcriptional response or the metabolomes of the animals in their ponds. But you can get a lot of useful information from doing that. And as scientists, we do have access to those methods and an increasing understanding of the value of those methods. So challenge number 2 is working out how best to deploy these new and emerging and next generation, whatever you want to call them, technologies, so that we get better understanding of what some of those more easily observable metrics and indicators actually tell us. So if we can see that the animals are behaving in a particular way, but we don't really understand what's going on, doing some real high-end science can tell us that they are stressed because of temperature, oxygen, heavy metals, whatever it might be, because we can read those sort of meta-omics data and get that information that you can't get any other way. But then we need to translate that back into making it more easy for farmers and other people to act on them. So the 2 challenges, one is to better understand what the easily observable indicators are telling us and what we can do about it. The second is, how do we deploy harder science to understand what's going on generally, and then take the simple messages of that back to the farms with confidence from both our point of view and the farmer's point of view that they are measuring something real and they can do something, something really impactful to improve the situation. So another aspect of, I think all of these things, One Health systems things, they rely on networks to actually achieve what we're trying to do. There's a lot of information out there. A lot of research has been done. There's a lot of local knowledge. but a lot of it is still dispersed and is not easily communicated or not communicated across the wide aquaculture landscape. And therefore it loses some power in that. I mean, if we had things that were more accepted, and I'm overstressing the point really, but because some of these things do exist, but if we had things that were more accepted and can be recommended as measures for improving things, and we did that in a way that was sort of very publicly visible, interacted with by both the scientists, aquaculture professionals, and the farmers, those networks would then become more self-supporting, more powerful, and more impactful. There are some out there, but again, there's a feeling that they are quite dispersed. This is an example of one that we set up 7 years ago, and to be fair, not much has happened with it. It was a good idea at the time, it's a good idea now, but it hasn't been animated. So my challenge number 3 to all of us is, do we think that this kind of network approach is one that is underutilized? Can we use it more? Is there an appetite from us and the stakeholders to actually pull these things together? It's a community effort, it would have to be, and it would need input from the farmers and everyone involved across the whole process for it both to be correct and powerful, but also to be implementable. And certainly for younger, earlier career scientists, I see this as a real opportunity in a way, because social media, these sort of online platforms and facilities are, people love doing them, and the younger generation seems to be better at them than the older generation. So there's a real opportunity for earlier career scientists to make their mark here and sort of pick this up and start building this network approach up. There's a lot to build on. For example, at CEFAS, we've got the World Collaborating Centre for Aquatic Animal Diseases, the FAO Reference Laboratory, for AMR, and one for bivalve sanitation. And I know, obviously, there are lots of these things about. So it's not about reinventing the wheel. It's about condensing things and presenting them in the most sort of attractive, interesting, engaging ways so that they have real impact on the ground. So something to talk about as we go through. But it does need a diversity of partners here. It needs academics, needs social scientists, needs economics, and needs people who can actually implement the stuff to sort of gain that trust and acceptance. An example of this is just a tender that's open at the moment from a program we've got at CEFAS as part of the Blue Planet Fund, which is called the Ocean Country Partnership Program. So this is to establish and coordinate an aquaculture network to deliver best practice, knowledge sharing, capacity building in Bangladesh. So along the lines that I've just been talking about. And the idea here, again, is not to do what we normally do under this program, which is to have a particular relationship between the OCPP program and a particular university or government department or research institute in a country. But actually to help build a network within that country, in this case Bangladesh, but the OCPP works in a range of other countries as well. So that network has a legacy and a chance of life beyond the OCPP program. And it also has that diversity of skills and ability to implement that those single bilateral relationships between the program and a single university would not have. In those cases, the funding ends and usually the project ends. This is a way of trying to give it a diversity of capability and a longevity that goes beyond the program itself, and then is a basis for attracting further funding. So it's a chance of getting a longer term program where a lot of these sort of aid type programs are short-lived, which is not to anyone's benefit. So that's available online on the gov.uk website if you want to look into it further and apply. Okay, so now on to the microbial section. So we've said a few times that the aquatic environment is a microbial soup, and that soup includes pathogens. It includes a massive diversity of organisms, the biggest diversity on Earth. I mean, the diversity of microorganisms in any aquaculture pond is bigger than the observable diversity in any in any coral reef, for example. And all of those organisms are sort of having some ecological role in there and a range of relevance to the aquaculture system, either as food for the animals in there, vectors of disease, they're agents of disease. They all have their own microbiomes and sort of host microbe environmental interactions, and we don't understand enough of this. What we tend to do at the moment is look for individual pathogens, normally in the animals themselves, sometimes in the water around them, which is informative up to a point. So this is a paper we published last year about using environmental DNA or environmental RNA for investigating pathogens in the environment. So in this case, an aquatic system. So the red dots here in this schematic at the bottom just illustrate a particular pathogen, and you can use these eDNA methods, and there's a whole wide range of them that I won't go into, but this paper does, to target those particular pathogens and look for them in the water, all the way through more and more animal-associated matrices to looking at them in tissues of the animals that you're interested in. And obviously that bit on the far right is more traditional molecular diagnostics, but there's a logical continuity in terms of eDNA detection, molecular detection, across that whole range there. So you can use that for individual pathogens. You can also use it for microbial communities to look for opportunistic pathogens, to look for metrics of microbiome health and microbiome condition. So a really powerful set of methods, eDNA, that we could use more in aquaculture. Again, it's not going to be an on-the-ground toolkit. It's a bit too complex for that, but it's certainly something that researchers can interact very positively with farmers to inform more about the system, to look into disease risk, both from the environment and from, for example, new aquaculture species being brought into cultivation. There are caveats, though. This is from some work we've been doing in Bangladesh over the past few years with Mafajol and his team, where we have been looking in a range of ponds. This is for monodon. And we've been taking an approach looking at using histology and molecular analyses of the shrimp tissues themselves. So that's the A here, animal. And then I is incubation, where you take the animals from the pond and put them in some clean water for an hour and see what comes off them and out of them into the water. And then you've got P, which is the pond water. So here are the pathogens that we found at the top. One of the beauties of eDNA methods is it can find pathogens that you didn't know existed. So this is a Bangladeshi, well, maybe not just Bangladeshi to be fair, but this is a new version of white spot syndrome virus that we found in those ponds. But these profiles are different. So for example, PMNV, we found very commonly in the animals. In fact, it was one of the most prevalent pathogens, but it never appeared in the water samples. So an eDNA assay for PMNV would be useless as a risk tool because it's not there. Other ones like white spot syndrome virus, there's a very good correspondence between what you see in the animals and what you see in the water. Appen, for example, is the other way around, where we found it a lot more in the water than we did actually in the animals. So that could be a very good sort of early warning of risk tool for Appen. So for each pathogen, the same eDNA approach gives you different kinds of information. You've really got to understand that to use these eDNA tools intelligently and usefully. But it's not just about these individual pathogens, it's about the whole set of microbes or symbionts. They're not all just microbes that are associated with larger animals. All animals have a set of symbionts with them. The animal and those symbionts is collectively called the holobiont, and we'll come on to that a little bit in a minute. But the pathobiome idea came around because we sort of increasingly realized that just thinking about one pathogen causing one disease is not accurate. It's not really how things work. Even when you've got a really strong primary pathogen like white spot syndrome virus that is causing disease, that has implications for the rest of the microbiome of the animals that can either mitigate a little bit the effects of the disease or amplify them. In some cases, it can give some kind of protection against it. In other cases, it can make it worse. But whatever, the rest of the symbiome changes when any sort of primary pathogen is involved. And sometimes diseases are caused by combinations of pathogens, not just a single one. And that's generally overlooked by, well, it is overlooked by all of the diagnostic approaches, the biosecurity measures that we take into account, and the way that we think about disease. So it's really important to think more holistically. Again, it's a systems, microbial, environmental, ecological approach that gives much more insight into disease and the susceptibility to disease. So that paper's been out for a while now in Transecology and Evolution. And this is, links us to the idea of syndromic disease, where we recognize that there's not just one pathogen. Sometimes there is, and we don't know what it is, and then when we find it out, like white spot syndrome virus, it doesn't really become a syndrome anymore. But a lot of things, like slow growth syndrome, the retarded growth syndromes in shrimp, don't seem to be associated with single pathogens. Things like EHP are very often involved, but they're not the single cause. So we need to think about agents that we know about as part of a pathobiome. We need to think about unknown agents. We need to think about the genetics, physiology of the host, the ecology and the environment. And there's a time and space element to all of this as well. And there's a range of different tools, and this is the important thing. Again, not just one tool, but lots of different tools need to be brought to bear to understand these systems. And ideally to understand a causal chain of events rather than just what's happening at one particular time. Because we said earlier, environmental quality leads to more parasite infections, leads to more bacterial diseases. These are a chain and cycles of events that we really don't have a grip on at all. And in order to understand aquaculture, we need to really get that grip. So this is a review we did a few years ago just about these suboptimal growth disorders in farm shrimp. There's a whole load of names for them. We just decided to lump them together into that title. And the important point here is this idea of a concept of individual pathogens as necessary but insufficient cause of disease. So you might need 5 different pathogens to sort of cause the disease or to make it manifest. If you've got just 1, 2, and 3, it's not going to happen. The disease won't be seen, but you do need 1, 2, and 3, as well as 4 and 5, in order for the disease to manifest. It's still a hypothesis at the moment, and it's not just the pathogens involved. As I say, it's the environment and the nature of the animals themselves, but it is beginning to give insight into these more complex diseases. Okay, this is the last slide, really. Just going back to this idea of a holobiont. So remember, a holobiont is any organism plus the smaller organisms that are associated with it. So that's bacteria, viruses, fungi, other small eukaryotes. And pretty much everything is a holobiont. Even an amoeba has a real large diversity of bacteria and viruses associated with it. Larger organisms have a lot more. And this is from a paper that we've got in press at the moment that is looking at how microbes can be used as bioindicators via eDNA methods into animal and ecosystem health. So a lot of bioindicators tend to be invertebrates, benthic invertebrates, for example, to understand the ecological status of the marine seabed. But microbes are massively more diverse, as I said before, and functionally massively diverse, and they offer a potentially really big source of environmental bioindicators, but we just don't know enough to do that. So this is an attempt, this paper reviews to what extent microbes are used, to how much they could be used, and what approaches need to be adopted to make them useful. And this particular figure from the paper takes into account the idea of this holobiont continuum. So you've got your holobiont that might be a salmon that's got a core microbiome and a more peripheral microbiome, and that forms a continuum into the environment through to microbial consortia and free-living microbes like the eDNA probing of the pathogens we talked about earlier. But a lot of work is done on looking at the microbiome of an aquaculture pond or another environment to understand the health of that environment, and that's good up to a point. But actually what we really should be looking at is where the microbes and the animals and the environment intersect, which is in this box here. Because at that point, they're all interacting, and at that point, you're going to get a much better proxy for what's going on in the environment as the whole than just looking at the free-living microbes or just looking at the health status of the host in terms of individual pathogens. So if we can use the whole microbial diversity associated with the host as a way of measuring the whole system, I think that would be much more powerful. It's barely happening at the moment. There's a few studies I've seen, none of them really in any of these aquaculture systems, but I really think this is a way forward. So that's it in summary, just for the systems part of it. We need to use ways of operationalizing One Health. These observable and easily measured indicators is, I think, a rich way of taking that forward. The development of local, regional, and international networks, I think, has got massive potential. And using those for knowledge exchange, skills sharing, and really importantly, implementation. From the animal and environmental health element, we've got this idea of symbionts, replacing the single pathogen agent traditionally considered part of the pathogen environmental triad. So this is the idea of the pathobiome. We talked about syndromic diseases, which are a systems manifestation of the system as a whole. We said that tools for understanding syndromic diseases and pathobiomes need to be really diverse in order to understand the system properly. Going back to some of the talks this morning and sort of thinking about the sort of microbial soup element of this, can we actually manage pond ecology and management from this microbial holobiome perspective? Even when we put feed in, that comes with a microbiome as well. If animals are feeding on organisms, phytoplankton, zooplankton in the system, they are eating things that already have a microbiome that is adapted to the stresses in that system. So it will be better food for them if only we can make enough of it. So going back to what you were talking about this morning, Jens, it's based more on the system itself with sort of minimal inputs from elsewhere. And then finally talked about focusing on this holobiont idea rather than looking at the environment or organisms for this integrated measurement of health of both the organisms and the environment that they live in. Okay, thanks very much.
Thank you, David. One quick question from the audience? If no one has one, I'll ask.
Thanks a lot. My question is aiming more at the part of the fish, the immune system. In one of my first lessons in aquaculture, I've been told that a fish is never sick. It's either healthy or dead. And in practice, this fits quite well, actually. So I believe that the immune system of the fish is actually key to all diseases at the end of the day, because as we've heard, most of the pathogens are there anyway. They just don't cause a disease as long as the fish is healthy. Well, what role, according to your understanding, does this immune system in the fish play? Is it possible to really prevent all diseases if the immune system is up to the mark, or will there still come diseases one day?
There will still come diseases because at some point that combined effect of pathogens, other microbes, and the environment will overwhelm the immune system, obviously. I mean, that's what happens. Yeah, I mean, the same thing's true of a set of birds. You know, they can either fly or they can't, and if they can't fly, they're dead. With fish, maybe they can be sick a bit more or compromised in some way. Yeah, we know that the microbiome of the fish is an important part of its immune system, so it has a really positive aspect there. But it can also have this sort of chronic debilitating effect that makes it more susceptible to, you know, some major pathogen that comes in that then wipes them out. So I would say it's much more of a continuum than that it's a live or dead model.
Thank you.
Thanks very much.
All right, our next speaker is going to meet with us virtually. It's Dr. Mohan from India, and I'm not sure if he's on now, maybe? Yep. His talk is going to be on integrating system thinking and observational epidemiology to better manage aquatic animal health. Dr. Mohan, can you hear us?
Good afternoon to all of you, those attending physically and those attending virtually this AquaSustain conference being held in Bangkok. Thanks to Andy Shin and Partho for asking me to share some of my thoughts on aquatic animal health with special reference to One Health issues. Today I'm going to just share a few of my thoughts around how systems thinking must be used and how observational epidemiology must be used to better manage aquatic animal health in farming systems. As most of us know, the science of aquatic animal health has progressed significantly over the past decades. Areas such as molecular diagnostics, molecular epidemiology, science of microbiomes have all advanced significantly. And there is also increased emphasis now on concepts like One Health, global food systems, need for food system transformation, etc., etc. So if you see all this, the main question is how should we produce food that is safe, that is nutritious, and that's affordable to millions of vulnerable communities, especially in continents like Asia and Africa. So this is where species like carps, tilapia, and catfish assume global importance, and also the need for injecting more aquatic animal health science into these farming systems. If you take the total aquaculture production globally and see where carps, tilapia, and catfish come, they are a major contributors to the aquatic food systems in both Asia and Africa. But if you look at the investments that's made with respect to research, development, vaccine development, diagnostics development in carps, tilapia, and catfish, which are affordable, low-value species, this is very, very, very small or insignificant compared to that of high-value species like salmonids and shrimp. So this is very, very sad because carps, tilapia, and catfish are very much needed to cater to the aquatic food needs of people in Asia and Africa. At the same time, if you look at the international and national investments on genetic improvement programs, dissemination of improved varieties and improved breeds, scaling programs, they're very well developed for tilapia, they're very well, for salmonids, fairly well developed for shrimp. But for tilapia, carps, and catfish, it's slowly gaining importance. You know about some of the important genetic programs for tilapia, for carps in many countries in Asia, and also for catfishers in some other countries in Africa. So when we look at genetic improvement programs, and then if you want to get the best out of the genetically improved strains, then understanding disease and generating scientific evidence around this to minimize disease-related losses is very, very important. Otherwise, many of the chronic low-level mortalities happening in carp, tilapia, and catfish farming systems largely gets unnoticed, unrecognized. Nobody gives importance to that. But we do tend to lose large volumes of these species because of chronic disease conditions. So understanding disease and genetic scientific evidence is very, very important. At the same time, for the improved varieties that are being disseminated and scaled across Asia and Africa for these low-value, highly affordable species, there is an urgent need to have a health management package or a package of practice to accompany these improved strains so that we can reduce the yield gaps and get the best out of the improved varieties that are being widely promoted so that it can really contribute to the aquatic food security for the needy people. So ultimately, what is required is how simple better management practices can be developed, how simple farm level biosecurity plans can be developed based on the risks identified, and ultimately contribute to the sustainability of these farming systems, which are low value species, mainly meant in many countries for domestic consumption, and the operations largely undertaken by small scale farmers. And nowadays, One Health is assuming a lot of importance. People are more concerned about antimicrobial resistance. People are concerned about foodborne illnesses, zoonotic diseases. At the same time, environmental health issues. Which type of farming systems has high footprint? Which sort of farming systems have low footprint? So people are more and more concerned about environmental health issues, at the same time human health issues, in addition to the health of the species which we farm. So One Health is really becoming very, very important. And in future, Asia and Africa will also be investing a lot of resources to get a better grip on One Health issues affecting those farming systems. If you look at the way the food safety is being treated in many countries in Asia and Africa, the regulators, the producers, the consumers, the value chain actors, they pay very limited attention to low-value species like carps and catfish and tilapia. But high value species like shrimp and salmonids get a lot of attention. So there are drug residue testing programs present in many countries. There are antibiotic residue testing programs. There are very well developed disease management schedules and programs for the high value species. But low value species meant for domestic consumption are largely neglected. And this is not really acceptable when we think of One Health and when we think of a major, commodity like aquatic foods coming out of carp, tilapia, catfish, and other minor carps and other smaller low-value species, which are taking care of the needs of people, marginal people in Asia and Africa. So there are two approaches. I think nowadays a lot of attention is being paid to those two things. One is the systems thinking approach, and one is observational epidemiology. The very fact that earlier approaches when we talk of disease was largely to look into single pathogens, diagnostics, understanding the pathogen, isolating the pathogen, characterizing the pathogen, and then trying to develop some management aspects or some treatment issues. But now it's no longer a single pathogen, single disease diagnostic issue alone. It needs to be seen in a totality of the various systems that is there. The systems would involve right from the entire value chain of the production system, right from the hatcheries to the harvest and post-harvest, the people involved there. So a systems thinking approach is very much needed, and a big shift is required when we talk of health management, especially for this low value affordable species. We should shift our thinking from a single pathogen diagnosis, treatment. Beyond that, we should think, I think, it's very important to have a systems thinking approach. Similarly, we are missing the potential value of observation epidemiology. A lot of important information can be gathered through very simple observations from the farming systems, which is largely going unnoticed in our rush to go just look for samples, look for pathogens, look for isolates, look for PCR positive products. So we are missing many things which have to be captured typically through using an epidemiological approach. So if you look specifically, what does systems thinking approach mean? As all of us know, especially carps and tilapia, and also to some extent catfish, the production systems are very dynamic. Polyculture is very prevalent. And the open and partially closed systems are very common. So we do not have 100% biosecure closed farming systems for these three commodities, which are very, very important for the future of the food security in Asia and Africa. So understanding these kinds of aquaculture systems and the polyculture system, how do diseases behave, pathogens behave in a polyculture system? How do host pathogen and the environment interact in a polyculture system? How does the microbiome dynamics affect the species that are being formed there? So using a approach where you think considering the totality and considering the whole system into account would be very, very important. And people have very clearly demonstrated that if somebody takes a very broad systems thinking approach, then it is possible to quantify risks to the farming system. It is possible to describe the different processes of the farming system. So one could very easily describe what are the different processes of a typical carp farming system in India, or a tilapia farming system in Bangladesh, or a catfish farming system in Nigeria. So a systems thinking approach is very important, and we should move away from straight away a diagnostic approach where collect sample, isolate a pathogen, characterize it, then think. No, I think we should move away from that and try to really understand what are the different processes in these farming systems. Through this, it is also possible to identify the pathways of exposure of our fish, maybe carps, tilapia, or catfish, to the different types of pathogens. Here again, it's very important to think and understand how ectoparasites, endoparasites, and various infectious agents interact in the aquatic environment, and then ultimately some infectious diseases taking an upper hand and causing chronic mortalities or acute mortalities or mass mortalities and spreading from one area, one farming system to another farming system. So if somebody takes a systems thinking approach, a participatory approach with the farmers and the extension service providers, it is possible to identify pathways of exposure of fish. Same is true with respect to identifying hotspots for disease emergence. Where can we expect some diseases to emerge? Is it this place or is it that place? It's easy. After COVID, people have very clearly seen the importance of sort of a systems thinking approach and how hotspots for disease emergence can be identified. For example, in COVID, the wet markets, how some of the slaughtering houses, how people can identify what are the hotspots for emergence of some dangerous diseases. So systems thinking will be more important than a pathogen and a diagnostic sort of a thinking. Same is true for antibiotic resistance. It's easy if you take a systems thinking approach to identify hotspots where antibiotic resistance could emerge, how it could emerge, and which kinds of bacterial pathogens the resistance could emerge. Ultimately, you can also pinpoint the roots of human exposure to this, either to the antibiotic residues themselves or to the the resistance, the antibiotic AMR type of thing. At the same time, epidemiology is a very powerful tool, but very few are making use of it, especially at the level of the farming, the systems, farming clusters, and the extension service providers. It's a very powerful tool because the farming ponds and the farming systems act as the natural laboratories for us. And the observations of the farmers and the farm level service providers is very, very important because they are the true researchers. They'll be able to see what's happening in the farming system. Are their fishes feeding properly? Are they gasping for oxygen? Are they swimming upside down? Are their bellies bloated? So a lot of observations are there which are of vital importance and which can give a lot of interesting ideas about the clinical science, about the pathology that could be already happening within the organ system of a fish, the manifestation of the functional failure of an organ in the form of a clinical sign. So observations are very powerful. Observations can give a lot of attention, importance to your sampling procedures and methodology, to your final, diagnostic decisions which you take. And many times we do miss to give importance to describe the circumstances which has given rise to a disease emergency or to an unusual mortality events. Normally we refer to it as case history. So giving attention to the circumstances under which a nasty infectious disease has emerged and leading to mortalities either in carp or tilapia, or, so this type of observations and trying to understand the case history from a population perspective, not in one fish, but in thousands of fish in a pond or in several ponds in a farm or several farms in a village or several villages in a district, keeping a population perspective and trying to understand the circumstances would be vital to design and devise effective health management measures. Same is true with clinical science. Many times we tend to neglect clinical science, but clinical science and the nature of mortality, whether it's acute, dramatic, chronic, low, high, these can give a lot of important information about the severity of a disease taking place within the organ systems of the fish. It can also tell us about the pathological changes taking place within the organ systems of the fish. So clinical science should never be neglected, and efforts should be made to really understand the relationship between the clinical science, it's linked to a function of an organ or a system, and also to the functional failure of that organ system, how it is manifested in the form of clinical signs. For example, drop seed condition, we see it commonly in carps and catfishes in farming systems. So it could be largely because of the functional failure of the kidney or osmoregulatory issues, all related to the problems associated with the fish. And then it could be an infectious disease, it could be a highly contagious viral disease. So the clinical signs are very important, and the nature of mortality is also important. And describing a unusual disease event taking place or a mortality event taking place either in a pond or a group of ponds or in a village or in a group of villages from a population perspective would be very important for anybody to really come up with good strategies, risk management strategies to either contain the disease or prevent it from spreading from beyond one unit to the next unit. So both the systems perspective, considering things in totality, and observational epidemiology, where we use the power of observations and the first-hand information, which is coming out of farmers and the extension service providers. Because when we as researchers go into that farm or the pond, it's already sort of, we are second stage. The first stage is actually coming out of the farmers. So that is very, very powerful, which is the first sight and the first set of ears and eyes on the ground in the field. So those observations will be very important for disease management, for One Health management, for identifying hotspots. So what is very important is how can we really stimulate and motivate farmers and service providers to be keen observers, to be keen in documenting systematically what they see in the form of pond record books or observation books. And to some extent, can they be trained for gathering simple samples, good samples, not a dead fish, but a moribund or a dying fish, which could be useful for series of your diagnostic approaches, right from pathology to PCR and reporting. Because there's no point in observing and keeping it to us. It has to be reported to the concern. It could be researchers, it could be scientists, it could be the government officers of the competent authority. So involvement of farmers and service providers is very important because they are the first to notice any issue in a farming system. And unfortunately, in shrimp and salmonids, this is very strong. In shrimp, it's largely strong because of the active involvement of the value chain actors, especially the feed people and the feed agents. But in tilapia, carps, and catfish, the involvement of these active service providers is less, and farmers also may not give much importance. So we lose vital data, vital evidence, vital science, which are not getting recorded. So whenever we think of putting up capacity building programs, I think a lot of attention must be given for inculcating observational epidemiology and also systems thinking within the capacity building programs, and training the farmers mainly how to observe, what to document, what to collect, what to report, whom to report. I think this will be a very good first step in these three major commodities to improve the biosecurity governance, to help improve the health governance, and also to contribute to this overall so-called One Health aspect of these farming systems. But at the same time, many times we get asked, what is the benefit for the farmers? We have to really consider what sort of incentives can be provided to farmers. And nowadays, a lot of real-time health advisory sort of apps are being developed. So we need to really look at to what extent these are really helping to motivate and stimulate the farmers and service providers to put in quality information, which could be used not only by researchers and scientists, but also from the national programs, national surveillance agencies, by the competent authority, so that the evidence in the form of data gets captured at the different levels, but very importantly at the level of grassroots level of farmers at the pond site, which is very, very vital for us as fish health and aquatic animal health researchers. So motivation and capacity building of farmers and service providers is very important. This is where I think ultimately what can be done to change the behavior, the practice, and the attitude of small-scale farmers who are occupied in farming of carps, catfish, and tilapia, which are a very important group of fishes for the future of aquatic food systems, for the future of aquatic food security for many millions of vulnerable communities in these two major continents, Asia and Africa. Because it's not the shrimp, it's not the salmonids, but it is the carps, tilapia, and the catfish. So to conclude, what I personally feel is there must be stronger attempts to integrate our learnings and our experiences, both from the perspective of systems thinking and also observation epidemiology. And if these two things can be sort of captured, integrated, then it is possible to develop what I call very simple, evolving, dynamic, contextualized, practical and cost-effective better management practices and farm-level biosecurity plans. So once we have this type of things contextualized to different farming systems at the level of villages or the farming clusters, we automatically take care of One Health issues. We automatically integrate your food safety issues, zoonotic issues, fish health issues, environmental issues. So ultimately, we contribute to the so-called one grand concept of One Health, where we're not only talking about the health of our carp or tilapia or fish, but we're talking about the health of the environment, we're talking about the health of the people who depend on farming and also consume this fish from a food safety perspective, and ultimately the health of the animal which we are farming, because ultimately animal welfare is also gaining a lot of importance, and it's very essential that the species which we farm are farmed in a very acceptable conditions, so that they do not unnecessarily suffer. So the One Health is conceptualized and captured in such a nice way if systems thinking approach and observation epidemiology at a very simple level can be cultivated amongst the farmers and the service providers. So with that, let me stop, and a big thanks again to Andy Shen and Partho for giving me the opportunity to share a few of my thoughts. And it's kept the presentation very simple, but I'll be happy to answer any questions or contribute to any discussions. Thank you very much.
For questions, for those of us in the room, we've got paper and pencil. Please write clearly, and we will post them to them, and then post them on the internet, the answers. Old-fashioned. And so we'll send you some questions for you to respond to in the next day. Also, thank you for describing my vet program. The real problem is actually who pays for the service, like the advice. Someone has to pay for that. So anyways, our last speaker is Dr. Melba Reantaso. Sorry, Melba. Everyone knows Melba. And I am very disappointed that you weren't able to come here because I really wanted to see you. But thank you for joining us virtually, and the floor is yours.
The invitation to participate in the AquaSustain 2024, and I will talk about addressing AMR. The invitation to participate in the AquaSustain 2024, and I will talk about addressing AMR and other sustainability issues through the progressive management pathway for improving aquaculture biosecurity. I'd like to start my presentation with a quote: "Whatever you do it wisely and think of the consequences." And while we are free to choose our actions, we are not free to choose the consequences of our action. As many of you are aware, the aquaculture sector is challenged by a myriad of diseases affecting the major farmed species groups: finfish, crustaceans, seaweeds, and mollusks. If we look at the chronology of disease emergence, this is how it looks like. From the 70s, there were few diseases of parasitic, viral, and fungal origin, such as Gyrodactylus salaris of salmon, MBV of shrimp, and EUS of finfish. Going into the 1980s, we saw the emergence of infectious salmon anemia and sea lice of salmon, a bacterial disease of shrimp, NHP, IPNV of tilapia and trout, and several viral diseases of shrimp, including the most serious white spot disease. In the 1990s, during the start of shrimp farming intensification period, vibriosis had impacted the shrimp sector, and viruses were still emerging at that time. We also saw how koi herpes virus first appeared and how it had later devastated the koi, carp, or ornamental sector. In the decade 2000, we saw the emergence of serious diseases such as APEND, probably the most significant non-viral disease of shrimp, TILV and VNN of tilapia, and few other diseases of shrimp such as IMNB and EHP. Unless biosecurity measures are put in place at farm and governance levels, we can expect more exotic, endemic, and emerging diseases to challenge the aquaculture sector. This is how the global distribution of diseases of aquatic organisms look like. If we look at the economics of aquatic diseases based on available literature, economic losses are high and estimated ranging from millions and billions of US dollar caused by either a single pathogen or several pathogens. These unmanaged disease outbreaks with high economic losses reflect an immature aquaculture industry. A maturing industry requires a focus on disease prevention and understanding of disease impacts in terms of burden and investments. The current approach to disease challenges needs to be supplemented with an economic dimension for improved responses and more efficient resource allocation. From the first estimate of 6 million tons a year in 1975, world aquaculture output had the invitation to participate in the AquaSustain 2024, and I will talk about addressing AMR and other 12.8 billion US dollars by 2022. By the 1990s, however, the emergence and spread of new and other persistent diseases began to cause concern to aquaculture stakeholders that these diseases were slowing the growth of the sector. Concerns focused on three issues. First is the increasing numbers, frequency, spread, severity, and persistence of diseases affecting both farmed and wild aquatic species. Second is increasing costs to the industry. And third is the time lag from detection of an outbreak to development and deployment of control measures. 3 or more years can pass between the disease being detected and the placement of control measures. There are many known drivers, factors, and pathways to aquatic disease emergence, which can be categorized into four. One is trade in and movement of live aquatic organisms. Two, knowledge of pathogens and their hosts. Three, aquatic organism health management and disease control. And four, ecosystem changes. This slide shows a causal diagram of disease emergence in aquaculture. It highlights the exposure pathways for the introduction of a new and the various stress-related factors at the right side, leading to the increased impact of diseases that are already present. These are the endemic diseases. Potential exposure pathways, if we look again at the left side, in farmed aquatic organism populations include, for example, introduction of internationally imported live aquatic organisms, seed or their products, new pathogen emergence on farms, risky husbandry practices, for example, using live feed that has not been health certified, and contact with infected wild populations. If you look at the right side, stress-related factors that may increase the impact of endemic diseases in aquaculture include changes in management practices, for example, increased stocking density, poor water quality, breeding species outside of their optimal or native range, selecting species or strains for growth over disease resistance, weather events or changes in water temperature due to climate change, and a stress-induced increase in pathogen virulence. These pathways and or stress-related factors may lead to the outbreak of an emerging disease in aquaculture. The consequences of an emerging disease are dependent on the time of detection and the effectiveness of response to the disease, which rely on sufficient capacities in surveillance, diagnostics, institutional coordination, research, infrastructure, biosecurity, and disease control measures. Let me now take you to this initiative that FAO and partners have developed called the Progressive Management Pathway for Improving Aquaculture Biosecurity, or PMPAB. It follows the principles of being risk-based, progressive, and collaborative. A good understanding of the relationship between the host, the pathogen, and the environment affected by human actions is key to the implementation of the PMPAB. So what is PMPAB? It is a new way to handle disease challenges. The PMPAB aims to enhance aquaculture biosecurity capacity at the regional, national, local sector, and enterprise levels. And to do so, it builds on two things: existing institutional and legal frameworks, capacity and appropriate tools using risk-based approaches, and public-private sector partnership. Second is it builds on resilience to the biosecurity vulnerabilities. Developed into two multi-stakeholder consultations and several technical working group meetings, and endorsed by FAO's Committee on Fisheries Subcommittee on Aquaculture during its 10th, 11th, and 12th sessions, the PMPAP is expected to sustain, first, a reduction in disease burden; second, an improvement of health at farm and national levels; third, a minimization of global spread of diseases; fourth is optimization of the socioeconomic benefits from aquaculture; sixth, an attraction of investment opportunities into aquaculture; and lastly, an achievement of the One Health goals: health of the ecosystem, people, and cultured organisms. In the context of PMP, biosecurity refers to the cost-effective management of risks posed by infectious agents to aquaculture through a strategic approach at enterprise, national, and international levels with shared public and private responsibilities. So what is AMR? AMR is the ability of microorganisms, bacteria, fungi, viruses, and parasites to persist or grow in the presence of drugs, also known as antimicrobials, that are designed to inhibit or kill them. When microorganisms become resistant to antimicrobials, standard treatments are often ineffective, and in some cases, no drugs provide effective therapy. Consequently, treatments fail, and this increases illness and mortality in humans, animals, and plants. What causes microbes to acquire AMR? Microbes become resistant to antimicrobials because of random mutations, and bacteria can also acquire the blueprints for resistance in the DNA or the genetic material from other bacteria. Sharing of the genes that confer resistance to antimicrobials is common among bacteria. When antimicrobials in humans or animals are used or are present in the environment, the microbes that lack the resistance are inhibited or are killed, and the resistant microbes are able to outcompete the microbes and become the dominant members of the population. The use of antimicrobials drives the selection for antimicrobial resistant population. In this slide, you will see the relationship between PMPAB and AMR. The PMPAB has four stages, and at stage one, biosecurity risks are defined. The first initial and essential activities include the development of a national or regional aquatic organism health strategy. You will see here the almost 17 elements of such a strategy, including a national aquatic pathogen list, diagnostics, surveillance, and use of veterinary drugs, and prevention of AMR. Pathogen list is an important requirement in order to determine what hazards country is protecting its sector from. Diagnostics, another essential element, is important as treatment should be based on correct diagnosis, and for bacterial diseases, antibiotic susceptibility testing is needed. In terms of our insights on adaptive management for bacterial diseases in aquaculture, I have divided them into two parts. First is prevention through best aquaculture, good aquaculture, and best management practices, enhancing gut immunity or influencing intestinal microbiome, optimizing immune status through vaccination, and use of SPF, SPT seeds, which means using clean seeds in the first place. The second part is control through responsible use of antibiotics and alternatives such as bacteriophages, probiotics, quorum quenching, bacteriocins, and medicinal plants. FAO has developed a 10-point biosecurity practice to reduce disease risk and prevent AMR. We are also initiating the development of technical guidelines on the use of SPF targeting small-scale producers. FAO's work on AMR is anchored on several instruments and frameworks, including the Code of Conduct for Responsible Fisheries, or what is popularly known as CCRF, FAO's Aquaculture Certification Guidelines, the FAO Action Plan on AMR, Blue Transformation, One Health, and the recently endorsed Guidelines for Sustainable Aquaculture. There are a number of tools to support the implementation of the National Action Plan on AMR. These include PMP AMR, Atlas, InFarm, RENEFarm, and the regional guidelines for monitoring and surveillance of AMR and bacterial pathogens in aquaculture. We are currently finalizing a 10-point checklist for AMU surveillance. FAO works closely with the reference centers for AMR and aquaculture biosecurity. The centers include Pearl River Fisheries Research Institute and the Yellow Sea Fisheries Research Institute in China, Nite University in India, CEFAS in the UK, and Mississippi State University in the USA. I'd like to inform the meeting that we are finalizing a book, Responsible Management of Bacterial Diseases in Aquaculture, containing some 40 bacterial species belonging to about 10 bacterial disease groups, 6 gram-negative bacteria and 4 gram-positive bacteria, including bacterial zoonosis. We are also organizing a 5-day hands-on training course on utilizing microbiome and genomic resources for understanding and mitigating AMR in the One Health context. This is in collaboration with one of the 5 FAO reference centers for AMR and aquaculture biosecurity. That is Nite University, located in Mangalore, India, and in collaboration with Indian Council of Agriculture Research. This will be held for 5 days from 18 to 22 November in support of the celebration of the World Antimicrobial Awareness Week. Thank you very much.
Thank you, Melba, and thank you for providing or highlighting these great references. That concludes this session on AMR, and I'm going to hand it back to the organizers for instructions.
So thank you so much. This comes to the end of session 2. So we will now have a QR code on the screen. We request all the participants to kindly scan the QR code. And this is for the evaluation form of all the presentations that has been presented so far. So your evaluations are important. So please take a moment or two to submit your evaluation for each presentation. And after you submit your evaluation, we'll have a short 20-minute coffee break. So you can have your grab a cup of coffee and just have a moment and then come back. And we'll have a big question workshop in another 20 minutes. Thank you. We will resume the workshop again in 20 minutes. Please grab